Hello everyone, my name is Jianyang from ECNU in China. Today I'm going to give an overview of the traceability analysis. Earth system models have been widely used to simulate land surface processes that drive climate change. Among these processes, the land carbon cycle is one of the most uncertain parts. As shown in the past three IPCC reports, there is a large simulation uncertainty on land carbon sink since 2001. The new analysis on the CMIP6 models also shows that this large uncertainty will still exist in the next IPCC report. So, one key challenge for Earth system model is how to reduce this large disagreement in carbon cycle among Earth system models. To tackle this challenge, we need to answer one question: why terrestrial carbon cycle is so different among the models? In the past 3 decades, we have seen an increasing number of model comparison projects or MIPS. Although those MIPS have led to a lot of publications, they are not designed to explore the causes of the model uncertainty. So, our question is, can we trace the model uncertainty back to the sources? The basic idea is that if there are some traceable components of terrestrial carbon cycle, then we can trace the model outputs back to the sources, such as the model structures, parameters, and external forcings. In fact, the model that terrestrial carbon cycle is traceable due to the fundamental properties. These properties are: first, photosynthesis is the primary carbon influx pathway. The second, the fixed carbon by plants are then transferred among different carbon pools. Third, the rates of carbon transfers are dominated by the donor pools. Fourth, the carbon dynamics of different pools can be mostly described by the first-order linear equations. These 4 properties have been adopted by most carbon cycle models. The above properties can be represented by this matrix equation. The right panel shows the box arrow diagram of the cable model. Different models could have different pool number and different values in the vector B matrix C, A, and C, but they can be unified by this matrix equation. At a steady state, the change in total carbon storage over time is 0, so we can solve this equation for getting steady-state ecosystem carbon storage, or we call carbon storage capacity. Based on these components, we developed a traceability analysis framework for terrestrial carbon cycle models. In this framework, the steady-state carbon storage, here we call XSS, is determined by 2 components. The first one is the net primary productivity, NPP, which means how much carbon is fixed by plants during a given time period. The second component is the carbon residence time, which represents how long the carbon stays in the ecosystem. The carbon residence time is further decomposed into baseline carbon residence time, which is affected by some parameters such as lead lignin fraction and soil texture. The other component is the environmental scalar, which are affected by climate factors such as precipitation and temperature. Next, I will show you 4 cases in which we apply the traceability analysis on terrestrial carbon cycle models. The first case is how the ecosystem carbon storage capacity varies spatially on the global scale in the cable model. We specifically focus on the difference between biomes. This slide shows how different ecosystem carbon storage capacity is jointly determined by NPP and carbon residence time. For example, in the right panel, the evergreen broadleaf forest, or BEF, have a very high NPP, but a short carbon residence time. The tundra biome has a very low NPP, but has a long carbon residence time. Some barrier biomes, such as the desert, has very low carbon storage capacity because of the low NPP and short carbon residence time. We further decompose the ecosystem carbon residence time into the baseline carbon residence time and the environmental scalars. As shown in this figure, the tundra and the evergreen broadleaf forests have similar baseline carbon residence time, but the environmental scalars are much smaller in tundra than evergreen broadleaf forests. The smaller environmental scalars mean the stronger environmental limitations on the decomposition rates of organic carbon. So tundra has much longer carbon residence time than evergreen broadleaf forest. This figure shows the variations of temperature scalar and water scalar among biomes in the cable model. It clearly shows that the spatial difference in the environmental scalars is mainly driven by the temperature scalar rather than the water scalar in the cable model. Because the cable model can open or close the carbon-nitrogen coupling, so we also studied how incorporation of nitrogen cycle can affect the carbon cycle model. The right panel clearly shows that the cable model simulates lower NPP in woody biomes, but shorter carbon residence time for non-woody biomes when the carbon cycle is implemented into the carbon cycle model. The second case is applying the traceability analysis to compare the carbon cycle of two models. One is the cable model, the other is the CLM-CASA model. As shown in these slides, these two models have very different structures of ecosystem carbon cycle. Because the values of the A, B, C matrices are so different, so it is very hard for us to understand why they generate different simulation results, such as the values shown in this figure. The traceability analysis shows that the cable and the CLM-CASA model simulate very similar global mean values of soil carbon storage capacity. The cable model has a longer NPP but lower NPP but longer carbon residence time than the CLM-CASA model. The right panel shows that the longer carbon residence time in cable than CLM-CASA model mainly results from the baseline carbon residence time rather than the environmental scalars. The third case is an application of the traceability analysis on the LPG gas model. In this case, we run the LPG gas model with 13 different climate forcings from the same 5 climate models. Because there is only one model, so all the difference in the carbon cycle is from the external climate forcings. We found that NPP is a key contributor for the variations of global carbon storage capacity in the LPG gas model under different climate change scenarios. The fourth application of the traceability analysis is to evaluate the impact of new processes on carbon cycling models. Here, we developed the TICO model to incorporate nitrogen cycles with 3 different schemes. As shown in the left panel, each nitrogen processes are simulated with different assumptions by different models. Although all the 3 carbon-nitrogen coupling schemes reduce the carbon storage capacity in the carbon-only version of TICO, the reductions were driven by different reasons. It suggests that adding the nitrogen cycle into the carbon cycle, which is now happening in the CIMIP 6 project, may generate many new uncertainty sources for the carbon cycle and climate change feedbacks. The above cases are based on the steady-state assumption, so you may ask, how to apply the traceability analysis on the transient dynamic of carbon storage? As shown in this equation, we further define a third term called carbon storage potential to allow the traceability analysis for analyzing the long-term dynamic of carbon storage. As shown by the right panel, we can see that the carbon storage potential varies over time. This figure shows the transient traceability framework, which further decomposed the carbon storage capacity and potential into their key components. We further developed an online traceability analysis system called TraceMe to allow the web-based evaluation of CMIP models. In the future, there are some other potential applications of the traceability analysis. Those applications could include benchmarking analysis, parameter sensitivity analysis, and data simulation. Many of these techniques will be introduced in this and in the coming units. Here is a short summary of this overview. First, the traceability analysis is helpful to explore the source of model uncertainty. Second, both carbon inputs and turnovers are highly uncertain in current carbon cycle models. There are also many new challenges or opportunities for the traceability analysis, such as how to develop the traceability framework to include nitrogen, phosphorus, and water processes, how to develop benchmarks for the traceability analysis, and how to link the traceable components to parameters. This is my overview of the traceability analysis. Thank you so much for your attention.